organization through single particle trajectories. He has published in a couple of years, interesting papers on how you extract information of what happened in bacteria. Please, Pietro.
Thank you very much, David, for getting me over. It's always a pleasure to come here to the Gatehouse Seminar Room 2. So I come into this problem from very much an experimental physics point of view, and my background is in soft materials, such as polymer solutions and polymer melts. So it's probably quite complementary to many of you, so please interrupt if the language I use is not clear, and I can't guarantee that I have answers for your questions, but I'll try my best. So I'll give you a bit of an introduction to the bacteria chromosome and why it's interesting for me. But actually, before I do that, I'll tell you about many other things we do in my lab just with this one slide. I run a fairly big experimental group where the unifying theme is doing experiments with optical microscopy so that we can image single cells and a function of biological cells. And the questions we have there, I guess, our biggest project at the moment is on understanding the synchronization of motile cilia in the airways and in the brains of mammals, where we have these carpets of active beating filaments, and it's not really clear how they synchronize together to form a collective dynamical state. So here we're doing experiments, and we're trying to do also simple models for the flow generated by cilia. For projects looking at lipid membranes, and this is both taking lipids from living cells and checking whether those mixtures are close to phase separation. Often they are, and that means that most likely the biology is exploiting the presence of this critical point for phase separation in order to create regions that have a different concentration in coexistence with the background. And then we have projects that are actually even more inspired by biological questions. For example, on how malaria parasites come out of red blood cells and then go on to invade other cells. So here there are problems about the elasticity of the cell membrane and how a parasite actually adheres and manages to get in. So obviously there's a lot of biological detail, but also some kind of soft matter physics going on at the same time. So I will also tell you there are two key people in the work that I described today. One is Avellino, who finished his PhD it's a bit less than a year ago with me, and we're still working on his data. And a current PhD student, Michael, in the lab now. And the collaborators, all this work is funded by HFSP, the work on bacteria that I present today. And the PI of the grant is my friend Marco in Paris. There's other two important collaborators, Bianca, also in Paris, who is a microbiologist, and Kevin, who's an expert in microfluidic devices, which we use to carry out some of these experiments. And we have a big network of other people who are interested in various aspects of this. So before I tell you about the bacterial kind of chromosome, I'll tell you about the big question that we actually want to try to address. So of course, you all know that the chromosome carries genes that need to be read out and by RNA polymerase, and then ribosomes turn them into proteins. And in the bacteria, there is obviously no nucleus. That's what defines a bacterial cell. So everything is contained in the same volume. It doesn't mean that it's perfectly mixed. On the contrary, the more people look, the more it's discovered that the chromatin is concentrated in the middle of these rod-like cells. It doesn't occupy the whole space, the DNA. And on the outside of this kind of concentrated cigar stand the ribosomes. And so there's a lot of spatial organization despite the absence of a nuclear membrane as you have in eukaryotes. Anyway, so the big question is whether, of course, there are transcription factors, which are proteins that bind to a little bit upstream of the gene and can upregulate or repress the expression of that particular gene. And a lot of work is done in biology to understand what's known as the transcription network, which means the the whole kind of graph of these interactions of transcription factors, which are themselves expressed, so you can make kind of dynamical systems out of this whole thing. And it's very much, in the mainstream, it's very much thought as a kind of a binary system where you then kind of assume transcription factors play an active role, a passive role. And you can go a long way to understanding what bacteria will do. However, everybody will recognize that this is not a complete picture in abstracting to a graph of transcription factor interactions. You lose a lot of the detail that you have in the actual system. And one big question is whether any role is played in the system by the state of physically compacting the chromosome. So this is also an analogy with what happens in eukaryotes, where there are histones that kind of structure chromatin, and other ways in which the chromosomes of eukaryotic organisms are basically silenced by keeping certain regions very compact. And that enables eukaryotic cells with the same DNA to perform completely different functions, say, in higher organisms. So our question already from several years ago, we've been thinking about this for a while, is whether and to what extent bacteria might be playing similar games as eukaryotes. And in essence, to then try to have a more complete picture of how the bacterium behaves, as well as the existing data on which concerns transcription factors for the most part. And it's known that there are perhaps about 10 proteins that are called nucleoid structuring proteins in a cell like the E. coli bacterium, which is a model system about which a lot is known. And they play different roles in conformation. So some of them are in this cartoon, like the red ones are causing a kink, the yellow ones are bridging, the other ones are also kind of cross-linking. And these things are fairly well known from the structural and molecular biology point of view. So what interactions these proteins will have with DNA helices. A more open question is what effect they have on the gene expression. Data exists, but often at a population level. So if you knock out one or other of these proteins and then compare the relative amount of protein of all the proteins or interesting proteins in those populations, then it's known that knocking these things out has a big effect. But it's difficult from population data, which is an average over a cell cycle and lots of other conditions, to actually have a very precise idea of what they're doing locally. Okay, so this is the big picture. Some of our ideas as we set off to work in this area, we have summarized in this review from a few years ago. I think David is making a much more complete review at the moment. We had chat about reviews.
It is included now.
Now this review is included in your review, so it's all reviewed. Okay, so the organism I want to discuss is E. coli. It's a rod-shaped bacterium that grows at its fastest. It can divide every 20 minutes or so if you give it its favorite sugars, or it can grow much slower if you give it less favorite sugars that has to metabolize through more complicated pathways. You can make it divide, say, every 2 hours if you want to. And when you grow it fast, it also, as well as dividing more quickly, it also creates longer and fatter cells by a bit. But say typically between here and here, if it's just divided, it could be 2 or 3 microns. And then when it grows and it's just about to divide, say like this one, it will be obviously twice as long, and then it chops in half. And it's fantastic what's going on in terms of duplicating the genome and making sure that half of it is on this side and half of it is on this side. It does all of this without microtubules, without spindle, without any of the machinery that are present in eukaryotes. It's actually quite an open question how it manages to very reliably get the 2 copies on either side. It's not particularly our question today, but it shares some of the polymer physics inspiration, if you want. So the people trying to work out segregation are also using a lot of polymer models, and also trying to understand how perhaps the presence of some of these binding proteins changes the polymer conformation and the entropy in such a way that it becomes actually favored to segregate 2 chromosomes. So aside from segregation, the chromosome is quite compacted in a system like this. If you lyse the bacterium cell wall, which we do in another set of experiments that I won't discuss, immediately the DNA swells to being instead of being confined in 1 micron by 3, it will immediately go to 15 microns, like forming a much swollen sponge, if you want, in solution. And that happens, we think, not particularly because you lose the wall, because actually the DNA is not particularly confined by the wall, but you're losing all the proteins inside here, which people would call crowding agents, and you're also eventually losing these proteins that are binding, which also maintain the chromosome compact. Okay, so people have been looking at bacteria for for a long time. There's a lot of structure, and also I've already mentioned segregation that happens in a cell cycle, which is kind of a dynamically organized process or emerging process. In terms of structure, there is structure at many lengths, and many groups have investigated quite deeply these various levels of organization. If at quite a local level, through kind of an accumulation of elastic stress, you get formation of supercoiled. There's a mathematical term for this. The DNA is already double helices, but if you super twist it like you could do with old telephone cords, then you would get the side.
Pin.
Yeah, that has a different name.
Okay.
In mathematical.
Writing.
The writing, yes. Yeah. So there is that, and then some of that is stabilized by some of the nucleoid proteins, because some of these are thought to act as essentially buffers that can maintain one region downstream supercoiled and prevent that twist elastic energy from propagating further. There is kind of the physics of this crowding, so what it means to have all the other proteins that are not actively binding to the DNA, but they're all agents, and they're bigger than water molecules. So the thermodynamics of this polymer embedded in a solution that has a lot of these proteins is actually quite complicated. And then there's on a much larger scale, people have started discussing an idea called macrodomains. This is particularly pushed by a French team of Espelli and Bocard. So they claim that in E. coli and probably other bacteria as well, there's an organization at the level of, say, a sixth of the whole chromosome, and they have evidence that the horizontal gene transfer and swapping DNA happens predominantly within these regions. And so another question is also whether, as well as this genetic definition of macrodomains, whether there's any other signature of macrodomain, if they're actually physically wrapped up as you can think of, for example, the DNA as a parallel chain model. Are these actually, do they look like stable parallels or not? So I won't go any deeper, but there's a lot of very serious work and evidence about many of these things. So the experiments we do are done by optical imaging, and obviously we don't resolve the DNA itself. We do experiments where either we grow the bacteria on a flat agar surface, and it's also covered by a glass cover slip, so you get for a few generations the bacteria grow and remain two-dimensional, and you can see each one. Or we grow bacteria in these very thin channels that are the same width as one cell. So the bacteria can't swim in this thing. They also can't swim there because they are stuck in the agar. But here they can't swim because of the walls. They can still have enough freedom to actually grow. And as they grow, they push any neighbor up, and any cell which is down here or up here gets pushed into a wider channel that has a flow and gets carried away. So the beauty of this type of device is that once you load it, you can then sustain this bacterial growth almost indefinitely. And you can have hundreds of these kind of vertical thin channels. So in your microscope, you can then kind of go and change field of view, and there's always bacteria growing. It takes obviously more effort to set this up than to set some of these up, but then it can be left to grow. We typically run these experiments for 2 or 3 days. And it's also possible to do media switches, so you can get a steady state, get the bacteria happy with one sugar, and then suddenly change the sugar, or suddenly introduce an antibiotic, or whatever other perturbation that you can induce by putting molecules in the flow much more easily than in the other device. Having said that, a lot of experiments I showed today are done here because this basically you can do this so quickly that the throughput of actually imaging new cells can be very high also in this sort of experiment here. Whichever you do, there are then you can automatically analyze these videos. There what we call segmentation algorithms can detect the edge of each of the cells. And furthermore, what you see in this image and in that image are these bright white dots. Those are the key of the experiment I'm talking about today. So we've taken strains. Actually, I'll come back to this image, but I think I have explanation here. So we've taken strains. This is the bacterium. This is the sketch of the chromosome, and a very short region with the PARS gene has been genetically inserted in this E. coli chromosome. And this PARS is a gene from a different bacterium. It doesn't exist in E. coli. And then there's a plasmid, which means a small circle of another DNA, which is also inserted in the bacterium. And this plasmid carries the gene to make a GFP protein, so that's a fluorescent protein. And it's a protein that every time it's made, it's also fused with a protein called PARB. Now this PARB protein binds with PARS. And in different bacteria, this is a way in which chromosomes are made to stick together. But this doesn't exist in E. coli. So when this protein is made with the PARB, the only thing it can do is diffuse around and eventually stick to PARS. Now, not only one protein is made, this plasmid remains in the bacterium and keeps making GFP. So actually what we then see as the bright GFP dots are assemblies of perhaps 100 GFPs. So we've now calibrated, and that's a varying number between 50 and 150 GFPs. So it's a fairly big kind of blob that assembles, but it assembles at one place on the chromosome, and we know where it is. And we actually have 27 of these strains where the PARS in each one has been inserted in a different place, and we know which is which. So then, when we look in the fluorescence microscope, we're shining blue light, we gather green light from the GFP, and each one of these dots so each bacterium either has one dot if that gene hasn't been replicated yet, or it can have 2 dots if it's being copied. It can be a little bit more complicated if it's growing very, very fast. A particular place on the chromosome could already have been copied for the granddaughters, so you would have 4 dots. But these experiments typically there would be 1 or maximum 2 dots in each bacteria. And then there's a technique which is fairly standard, and lots of people use in different ways to localize the position of these dots with very high accuracy. Is everybody familiar with it? Should I say anything about this? Is it interesting or not really? Okay, say. So, a Nobel Prize was given a year or 2 ago for super resolution microscopy for techniques also similar to this. And the key idea with super resolution is if you're actually only caring about where an object is and not actually resolving its shape or whatever, then you're not limited by the resolution limit of the optics lens. You're actually limited just by the number of photons that you're capturing. So, because if each of these you can think of as a point-like source and it's emitting photons at a certain rate, the more you accumulate statistics of where they land in your camera, the more precisely you can tell where that point source was. So this is a regime that many people call photon limited in terms of resolution. So you make some compromise about how long you keep your exposure open on each of these dots. Obviously, if you keep it very long, you will have high precision, but if your object is moving about, you lose information about where it is over time. So you trade off the number of photons, which is your precision, to how many times you want to take a photograph. You also have to trade off other things, which, for example, this GFP, every time you're exciting it with the blue light, there's a chance that you will make it undergo a process called photobleaching, where it then stops emitting, it undergoes some chemical change. So if the more you image these, the dimmer they become. So you also have to trade off how many photographs you want to take in total, because once it's photobleached, they will not come back and give you any more light. Okay, so there are some experimental trade-offs, but in essence, we get the position over time of each of these dots with a precision which is about 20 or 30 nanometers. And then if I plot x and y of a particular dot, I will get something that looks a bit like a localized random walk.
So what was that bar?
This, I think, is 5 microns. Scale bar. Which bar, sir?
On both pictures.
Pixelator one. It's yeah, I think this is also 5. It's possible that it's 5 microns, because essentially the dot will look as big as what's, no, maybe this is 1 micron, because the dot will look as big as what's known as point spread function. So, and we're using quite high magnification, high numerical aperture objective. So the point spread function would be, of these, should be around 250 nanometers. So I think this will be 1 micron. But this is probably 5 because the bacteria are around 3. So, certainly in the soft matter physics and the rheology community, people have done a lot of tracking inclusions in materials of which you want to know the viscoelastic properties. And the measure that everybody does first of any kind of random walk is what we call the mean square displacement. So you look at the position at a time t, you look at the position at time t plus some t naught. Actually, this obviously I got the formula wrong. So if this is t, this is t naught plus t, this should be minus t naught, and then it's averaged over t naught to give you t. Apologies. So anyway, take a difference, you square it, and then you average over time, given a lag time of which you did the position difference. And a lot of kind of viscoelastic materials will have power laws as a function of the waiting time, and then of course if your thing is a random walk to normal diffusion, then your power law will have exponent 1. If it has exponent bigger than 1, goes super diffusion. If it has less than 1, subdiffusion. And a lot of some of these experiments have also been done in eukaryotes, and also in common here, all of these motions are subdiffusive. And that's true also of whatever you do in a polymer solution, you will get subdiffusive motion of tracer particles in a polymer solution, either diffusive or subdiffusive. Okay, so these experiments were done first in Stanford and published in this paper in the group of Julie Thériault. And we started doing our things more or less when this came out. And what they showed were mean square displacement plots. This is the delay time that showed power laws with exponents 0.4 and a huge spread here in the coefficient, but not a very huge spread in the exponent. I mean, these are all kind of 0.4s. We got our strains from the Espelli lab, and they were using those dots to look at things which were much longer. So their key question was this about the segregation. So they were looking at the position of the dots over the cell cycle. And here also, this other group have looked at dynamics from 1 second to 100 seconds. So we had, well, we thought our methods were good enough to essentially push to shorter timescales by 1 decade over here. And the reason to try to push the experiments over here is that we wanted to look at what we thought would be really kind of fluctuation behavior, and not be confused by any of the segregation or the bacterial growth, which are processes which, if you think of it, are super diffusive. Because the growth of the bacterium will actually give you like a drift velocity. And the same, the segregation. If there's a there's a movement of roughly 2 or 3 microns every 20 minutes that corresponds to a speed. And in a mean square displacement, that will have an exponent 2. So if we're after carefully looking at exponents which are below 1, we have to be careful not to cross over into another process which is completely independent and has a higher exponent, and at some point will take over and move your dot. Anyway, so we tried to push towards faster times. Okay, so we did estimates, and we decided we want, so this would be the exponent 2, and at before 100 seconds it becomes a bit dangerous to look at the data here, because of this is just coming from the estimated growth of the bacterium. So this is a summary of a lot of data. We pushed below a second, as I just said, and these power laws hold the same as in this decade here, between 1 and 10. They also hold down towards 0.2. We looked around the chromosome, and the first thing we noticed was that the data around this origin of replication, that's where the chromosome begins to get copied, and then you get two copies of this, and the replication forks walk along both. There are 2, so one each side, and they reach this terminus region, which is the last to be copied. These regions fluctuated more than these regions, and by quite a lot. We then had big discussions about the exponent. So these lines are 0.4, the black, and 0.5, the red. And for a while, I tried to convince my students that actually this was a 0.5, which we could explain very easily. But it turns out it's actually 0.4. The black is what happens if you cross-link the DNA, and it's a measure of essentially our precision in localizing the dot. So the fact that these data are higher than the cross-linked one, it gives us confidence that there's no issue with the precision of measurement.
Why should it curve at the end of the cross-linked one?
So these are vibrations from the microscope, for example. It curves upwards a bit. If you take the square root of this number in microns, it's a very small number. So these are very small movements, even the upturn here. Okay, so the exponent, well, when you look at a lot of tracks, so in this histogram there are 65,000, and then we kept on collecting stuff. I think we now have 350,000 tracks. It's really peaked at 0.4. You couldn't argue that this is a polymer Rouse model. A simple Rouse model would have 0.5, and for a while I was hoping we would just get 0.5, but no. And also that original parallel paper also had a histogram with at 0.4. And that 0.4 exponent is quite stable across the whole chromosome. The prefactor, so what was giving rise to this change here has a roughly linear change from origin, which is here, down to terminus of replication and back up to origin. The chromosome is a ring, so it's quite symmetrical if you go on one side of the ring or on the other side. And we then invested a little bit of time, so we had lots of tracks, and we could bin the tracks in different ways to try to see if we could single out some correlations with this effect. The best thing we found was that if we, so this is a quarter of a bacterium. This is the plane of mid-cell, and then there will be the other pole up there, and the other piece of the bacterium here. We've remapped everything on a quarter because it should be symmetric that way. And this is a heat map for where you expect, well, not where you expect, where we find the probability of finding that dot inside in a given place inside the bacterium cell. And then, of course, it's 3D. Also, it's a cylinder, so everything is projected down onto a plane. So this terminus region, the last piece to be copied, has a much higher probability of being at mid-cell. That's because as soon as the bacterium starts making the 2 daughter chromosomes, it manages to put the 2 new origins on either side, and the terminus gets pushed in the middle, where it's going to be until it's finally all copied and it gets chopped up into 2. But as soon as when the bacterium is just born, then it's not like that. The terminus is on one pole and the origin is on the other pole. So you have some chance of seeing the ter also near the pole or anywhere else. During these times, the terminus is actually moving towards mid-cell. So what we noticed, which I thought was interesting, is that if we looked at this very fast jiggling of terminus when it was not at mid-cell, then the jiggling was actually the same as the other jigglings from the other parts of the chromosome. So that's what this graph is saying here. We're looking at a particular lag time, 10 seconds. So it's a position on those mean square displacement curves. And we're comparing the chromosome positions. And ter, which are these yellowish, greenish, they are quite close to the other lines if we are away from mid-cell. And it's really at mid-cell that they then drop down. And the reason why they then matter a lot in the average is that when you do a time average, well, most of the time ter is at mid-cell, so it's actually these low motilities count a lot in the time average. But this is saying that there's probably nothing in terms of the kind of the gene sequence which is playing a strong role. It's actually something which is happening when the ter is at mid-cell that kind of confines the fluctuations much more.
Question, is that because its mid-cell is sort of denser or something like that?
So
or is it the folding of the DNA when it's over there, it's sort of folded?
Yeah, so that we are now exploring, because as you correctly are thinking, there could be a physics reason, perhaps the same reason that is causing the segregation, then causes that midpoint to be more physically constrained, so without any protein involved, perhaps. Or it could be a protein. And there's actually one protein called MATP, which is known to have a very strong affinity for this ter region, and it's one of the nucleolytic structuring proteins. And that MATP is perhaps actually binding and holding things there, and it might also be helping the segregation of the chromosomes. So it is possible to grow the cells without MATP. They're less happy, but it's possible, and we're now doing these experiments without MATP. So hopefully I'll have a better answer soon.
And you have you only have the ter at the point 4.
This one, yeah. So this is a ter just born because it's really close to the pole.
That's like one measurement of.
It's probably very few measurements, and for some reason it's not jiggling very much. But maybe it could also be that it's appeared here, but the cell wasn't fully divided. It's very few data points. I would make much more of this part. So okay, so the whole distribution shifts. The REs move more than the ters. It's just another way of showing you what I've already shown you. Okay, then we wanted still we hadn't really addressed this problem. So we also had this huge spread. I mean, this is a log scale. You have like almost 2 orders of magnitude of motion for little dots that are supposed to be the same on the same place on the chromosome. So we were worried about that, and what we saw was that there was a very strong correlation of how much the dot moves as a function of how bright it was. This hadn't been discussed by any of the previous papers. So here kind of enters a slightly technical thing. So maybe if you want to think in terms of maths, you can switch your brains off. But the next time anybody shows you dots in any talk, you can remember this artifact and start asking questions if you want to. So, okay, there are people working on systems where the dots will have all the same brightness. And that's true if they manage to make a fixed valency, fixed number of GFP molecules. But in many other systems will be like ours, they will not have a fixed number. The GFP will polymerize, and in different cells, you will end up having quite different number of GFP because of stochasticity in the production of that GFP. And that's what happens certainly in our system. So we have a wide distribution of GFP number, and we find that it correlates strongly with the motion for everything else being equal. So the brighter the dot, the less it jiggles about. So one thing we did was we convinced ourselves that this was really an effect of the number of GFPs, and not an effect of say precision coming from perhaps a big dot being measured better than a dim dot. We did that by just doing, say, videos on the same cells, but with different amounts of incident light, which then gave us different amounts of emitted light, because the two are proportional, and checking that we could then rescale all the, okay, this is the rescaling. Apart from the very, very dimmest, which then really run into localization problems if you don't have enough photons coming out. All the other excitation lights, once rescaled by how much excitation we were putting in, gave us the same mean square displacement. So this then really means that it's a physical thing, that the brighter the dot, the more GFP it's got, and the less it's moving. So then that points to, okay, that can then be, if we take the motility at say 10 seconds, actually this is the motility of 1 second, which we baptize d up. Because then independent of your exponent of the random walk, this has dimensions of micron squared, so it's kind of convenient. So this thing, as a function of this intensity, is itself looking a little bit like a power law. So here there are, from there to there are 2 decades, from there to there a bit more than 1. So it's going roughly down. This part's like -1, this part's a bit less. You can these are just guides to the eye in terms of power laws. You could think of fitting everything and it would be something intermediate. And in some sense, we think that buried here might become some real physics. What sort of physics? We don't know. We also had a chat with David a few weeks ago about this. It's possible that this is essentially the physics of a solid inclusion in a polymer matrix, and that this scaling of this intensity is basically changing the size of the inclusion. But the inclusion is not just a kind of a dumb inclusion, it's actually stuck to a particular point on that polymer. So this is something that this can be treated, or one can try to treat it in terms of simple models and see if anything kind of like these exponents comes out by doing that. What's interesting is that in all of this thing, the exponent itself remains 0.4. So the exponent doesn't change, but this prefactor has quite a clear experimental signature. We have done now the work of changing this intensity to actual number of GFPs, so we know how big the inclusion is in terms of a physical size. And this is the coefficient. Okay, this is so all of the stuff I showed up to now is published. I don't know if the references were there, but I can email them to you if you're interested. This is a slide of non-published stuff. So one way in which we're pushing this work is to see if this fluctuating motion of the chromosome changes when we mistreat our bacteria. And one thing we're exploring is putting antibiotic drugs at a concentration that doesn't kill the bacteria. So we chose half of the inhibitory concentration for each of the drugs. It's something we had to work out how much to put. And then basically the bacteria are slowed down and affected by the drug, but they're not dying off. And we looked at the amount of motion as a function of time, where now this time here is minutes. So these are 2 hours. So we're still making our very short, fast videos, but we make a short video, then we wait 20 minutes, we make another short video, then we make another 20 minutes. So that's kind of these individual data points are the same bacteria colony, but we're not photobleaching them very much, and we can image them again after 20 minutes. And we're seeing results that we don't understand. This is a locus near origin, this is a locus near terminus, and they do something similar. Basically, already the first imaging we do, which is already after 20 minutes of antibiotic, that there are 2 drugs that have reduced these fluctuations by a lot, and they are vancomycin and ciprofloxacin. And another drug, rifampicin, hasn't reduced things very much. The blue is no antibiotic. And then over the 2 hours, the bacteria seem to manage to kind of compensate for the presence of these drugs, and everything goes back to being quite similar. The most striking thing we don't understand is that we thought the rifampicin, which is a drug that affects the ribosomes, we thought would have some effect on on the crowding around the chromosome, and also perhaps on the on actual structuring, because the ribosomes are actually doing their thing quite close to chromatin. Whereas vancomycin is a drug that affects the cell wall, and we thought it wouldn't have an effect. And they've come out swapped around. So the drug that we thought would be closer to the chromosome is not making a big effect, and the drug that's affecting cell wall is. Now again, here we're discussing in the lab all kinds of reasons. There could be a physics reason, there could be a biology reason. A possible physics reason is maybe as we hit the bacteria with this cell wall drug, we are stopping the elongation of the bacterium, but we're not stopping any of the processes inside. So it's going into a slightly kind of overcrowded state. That results in a reduction in motility. That's my favorite explanation. My student, who is a biologist, has all kinds of biological explanations in terms of which molecules are actually affected by this drug. And until we do more experiments, we won't know if it's physics or biology. Okay. How am I doing? Keep going for 10 minutes?
Sure. Go ahead.
Any questions, Absalom?
Yeah, so in the previous picture, so I mean, because you kind of take videos after 20 minutes. I mean, doesn't that introduce quite a lot of error in how you go from the one dot to the other?
So we don't track the dot across the 20 minutes.
No, what I mean by dots, I mean in there, so you have like 2 data points, yeah.
Yes.
And my understanding is that the time that for 20 minutes you're not really doing it. You're not really doing anything. So I'm kind of wondering, and that's why we kind of see these kind of lines.
Yeah.
But how do you have a feeling of how big or how small the error is from the fact that you're not keeping track for 20 minutes? If I understood correctly, you don't want to keep track for 20 minutes continuously
It will
because
be too much light.
it would be too much light.
Yeah.
But then, I mean, when you kind of start, when you see it again, how do you know where it was before? Sort of.
Yes. So we don't do a correlation of the position across the 20 minutes. At each of these 20 minutes, we take probably 200 quick images, and we do our analysis of fluctuations across those 200 images.
Okay.
And then that's redone here, and we don't go across.
Okay.
But it's the same. So experimentally, we have many little colonies of the bacteria growing, like I had it in the first image, and the microscope is taking these videos on many of the colonies. So thousands of cells, thousands of dots are averaged to then make each of these motility data. So then comes there's an actually slightly tangential question, which I hope will be of interest to some of you. We have so many tracks, like 300,000 tracks that we can actually dig in and go and see if there's any rare events inside these tracks. And this was a bit inspired by this work by Nancy Kleckner in Harvard, where they claimed that over quite short time scales, so over 1 minute, a lot of like large-scale DNA motions could be seen across the bacterium body. And so we thought, well, if these things are happening, then maybe some of our tracks might be showing some strange effects because of these kind of big waves. So we looked, and we set some criteria to go and look for particularly elongated tracks. So our tracks, as I've said, they're made roughly of 100 or 200 frames at 0.1 second intervals, and a lot of them are basically quite confined random walks, but some of them are elongated. So this is a little zoo of elongated ones, and you can see elongated can be quite elongated. So these are very short. They are only 20 seconds long, and in 20 seconds, this dot has done a third of the cell length, so a micron in 20 seconds. So if you look at that elongated track again, one of the elongated tracks, like it will look very long. So here we could discuss this in terms of mean square displacement, and when we do, these things have an exponent of about 2, that they really look like, they have some noise and some jiggling, but essentially they have like a velocity as well, they are driven. So we also sometimes analyze these just in terms of velocity, so we kind of fit this within an ellipse and we take the major axis and divide it by the time to get a bit, what we call drift velocity. Or in other graphs that I will show you, we plot something called the mean absolute displacement, which is basically scales like half of the mean square displacement. This is because my PhD student was a fan of metrics, and we've ended up with plots plotted in lots of different metrics. But essentially, these are elongated, and they look like driven motions. So in terms of this, so this is the square root of the mean square displacement, and those elongated ones go linear in this representation, and the normal subdiffusive ones go like square roots. So this is the normal one, this is the elongated one. So that question was, we see just a few of these out of a gigantic data set. So we see between, well, just a few percent of all the tracks. Are these strange ones still something you would expect in the tail of distribution of random walks or not? So the simple thing we could do as physicists was just to set up a fractional Brownian motion simulation where we matched the exponent to be the mean exponent of all the tracks and the prefactors as well. Basically, we try to match the mean behavior of the population, and then we looked at how many tracks had the special features from this well-defined stochastic numerical model. So when we did that, okay, this is now plotted in terms of this elongation divided by time, so it has units of velocity. There's a distribution for how many times you expect this thing to occur. And the simulation are these red crosses, and this is their distribution. So you get obviously very few very elongated things. The data, so all of the data without any selection are these dots here. They have a much fatter tail up here. And if we set some criterion for the particularly elongated tracks, which are the ones I've been showing you before, and we remove them, then the data really falls on top of the fraction Brownian motion simulation. So that told us, but I don't know, maybe people here are actually much more expert about these things, that there's a small subset, this few percent of tracks which are not behaving as the mean behavior coming from fractional Brownian motion. So that same thing affects the mean behavior a lot. So the data curves up in the presence of these slightly elongated tracks because they really move a lot more. Even though they're a small fraction, they move more. And if you remove, again, the ones that have slightly higher drift velocity, okay, so these are actually, the yellow is the ones with the higher drift velocity. And the more, if you remove these guys and you keep only the ones that are below a certain threshold, then your data falls onto the 0.4 line. Okay, here it says 0.2 because we square rooted it more and more cleanly. So these things, we started calling them snaps amongst ourselves because snaps was already the term that the Kleckner lab had baptized, and we saw that they happen most often along the cell body direction, which is perhaps not so surprising. We checked whether they were happening, when they were happening relative to the fact that a particular point of the chromosome will get copied. So these replication forks are starting from the origin, coming along, duplicating that gene, and then going on afterwards. And there's a question whether that process might be accumulating stresses and then perhaps relaxing stresses. But it doesn't seem like that. The this fraction of the elongated steps it does have perhaps a singular event. This is time measured from when that position gets copied. So it does perhaps have some correlation with being copied. I mean, it seems to do, but it doesn't go to zero away from those events. So it's not. That doesn't seem to be the event itself causing the big movement. And two slides. Okay, and I guess that's all we've done with the snaps until now. They happen. Okay, what the plots above are showing us that they happen also for faster growing media or for slower growing bacteria as well, which is interesting because this very fast one with the casamino acids would have more origins, for example. So then, I don't think I wrote anything on these slides, but my favorite interpretation, again, in terms of physics, is the idea that probably this segregation motion that divides the two chromosomes, it's not a particularly smooth process. It's a process where the polymers will get entangled and will be accumulating stress, and then probably randomly at some point, the stress reacts away a bit like an avalanche, and these will be intermittent avalanche-like things that will show up as big displacements, big driven relaxations in the chromosome. This is a summary. I've shown you this very short time, the shortest that is currently possible motion of bacterial chromosomes. There's a very open question. Okay, maybe here I can spend a minute. There's a very open question about the 0.4. It's an annoying exponent because it doesn't come from any of the simple polymer physics that is known to us. In the early Terio papers, in collaboration with Andy Spakovits, who's a polymer physicist, they proposed the 0.4 as being essentially an analog of the 0.5 you get from Rao's model, but for a polymer which is fluctuating in a viscoelastic background. So if they said, well, if the background has a viscoelasticity with power law exponent 0.8, then the Rao's model machinery will cut that exponent by 2 and it will give you 0.4. But that just moves the question, okay, why is a 0.8 exponent in the cytoplasm? Which is actually a more difficult question because the cytoplasm has to respond to things like osmotic shock and crowding, etc. And we see a 0.4 which is very, very robust. It would be a bit strange for just protein crowding to come up with a robust 0.8 viscoelasticity exponent. So that, I mean, perhaps Spakovits is right, or perhaps there's a more kind of an explanation that relies more on the polymer, and that seems to me it would then be more robust to perturbations. In terms of our big question that I posed at the beginning, where there's another PhD student working on this link between these motions that we see and the actual gene expression. The hypothesis, and we hope, because it would be interesting, would be that there might be less gene expression from the points in the chromosome that are able to move less, because we would then say, well, they're more compact. And that would be similar to kind of gene silencing in eukaryotes. But we're actually still building the data set, so that's all speculation at the moment. And I told you we are doing the experiments knocking out MACP and also some of the other NAPs to understand chromosome dynamics. But we were actually slowed down here by the very pragmatic problem of that size of GFP question. So we've had these strains for a while, but until we understood properly, and to some extent we still are, the effect of the size of GFP, then it's not possible to compare strains that are dramatically different, because in a strain that is suffering more because of the absence of this, much less GFP will be made. So then the motility you measure there is very different from the motility in the happy wild type, but not because of any interesting polymer physics, but just because the dots are all kind of much smaller, for example. So even kind of the boring kind of amount of light from the dot is actually important to go and ask more interesting questions. Okay, so I've summarized that, and thank you very much.
Some questions? Let's start with
back to the thing with the fractional coordinate motion.
Yeah.
I didn't fully understand the no, this one later on when you, yeah, the previous one. I didn't understand the rationale of why you kind of throw away the data. So the blue data are the actual data.
Yes.
Yeah. So that would imply that in the actual data you have more trajectories doing long excursions.
Yeah.
But I didn't understand then why you're throwing away some of them. What was the rationale?
No, we're not throwing them away. So we isolate them, and they're the ones we have studied in terms of when they happen and which direction they have. So we've characterized this. I think what I'm showing with this graph and also with this is that if I take them away, then my distribution, both in terms of this distribution and in terms of what mean square displacements I get, go back to being the simple power law, which is not so surprising because I've taken away the ones which don't match with fractional Brownian motion with an exponent of 0.4, and so then they look like the simulation when I do any of the other plots.
Yeah, sort of following on from that, I mean, what should I say? If these snaps are kind of trajectories and unusual features, it strikes me fractional Brownian motion is more kind of like a smooth. I mean, the trajectories are kind of smoothly different from each other. Whereas if one looked at Levy process type of thing, then that it seems to me they're a little bit more intuitive. That you could have sort of unusual trajectories that you could condition on and get something for the unusual trajectory. So I'm sure you must have considered alternative models to FEMs to explain this, and they didn't work as well.
We didn't actually do too much. So what you're saying is that we should probably consider other, I would say, null models in the sense that they don't have very strange biological active processes, but they would actually at the same time have a good, well-behaved power law, but allow for strange long tails.
Yeah, but it's the usual. The physicists are always, arguing whether it's fractional Brownian motion or Levy walk or whatever it is. In some cases, it's one or the other.
Yeah, no, that's the kind of thing we haven't explored other models because we came into this with a polymer physics mindset, and it makes sense to look at the smooth models. Yeah. The Levy flights. I don't know.
Especially if you somehow think these snaps are somewhat punctuated events, that might make more sense.
Yeah.
Okay, so I have actually some questions and some comments. So we analyzed many tracks, not on this system, but on yeast. And we also found, my collaborators that did experiments and we analyzed the data, we also found a large spectrum of alpha.
Mm.
And so I saw that reducing this to 0.5, I understand that you have this beautiful peak. Actually, why not using the entire spectrum and say that actually it might be possible that some that are living at 0.2, 0.3 are actually a representation of some heterogeneity of the nucleus, of the organization, of the chromatin organization. And that's why at that time we built this, beta polymer model to say how not the explanation of Spakovich, but the explanation that you have this alpha because you may have longer interactions along the chromatin. And so keeping this heterogeneity, as revealing the heterogeneity of the local organization of the chromatin. So this is one point that maybe,
You think it's just like a multifractal framework you're thinking?
No, I don't think about multifractal. I think about the fact that the chromatin can be heterogeneous at some point. If you have the
so
same,
just some regions, some alpha, some other regions, another alpha.
yeah, and even the same region can have possibly different alpha because at some point of time it might be organized differently. It has a different local structure. Sometimes you walk alone, sometimes you walk with your dog, with your family, and it affects
So time heterogeneity is what you're saying.
time heterogeneity because this is not exactly at the same moment of time. And a lot of things can happen. So this is one thing. And second, I have to say that I agree with you that we looked also at the reason when you look at the motion with a drift. So we could not give any interpretation of the data we have with this anomalous fractal Levy flight motion. It was not a power law decay, it was an exponential decay. And this is also closely related to Brownian motion, and Brownian motion, again, on a polymer. That is, if you think about doing a simulation where you have your polymer and you look at a single bead. Now, if you hold your polymer, if you push on the polymer, you have a force. And this force is going to propagate through the springs. So you still have everybody doing Brownian motion, but still, you generate a force. And if you wait sufficiently long enough, I mean, so that the force that you're pulling here affects the motion here, you may end up, I mean, we did some of the simulation like this, but not in that context. We looked at what happened when the telomeres were attached to the boundary, and the boundary, as you know, somebody showed last week, can also oscillate. We saw how this affects the motion, and it does. As you said, when you have a combination of Brownian motion, which is square root of delta t, and deterministic drift, which is delta t, I mean power squared everything, then indeed the two are mixing together can affect the exponent. So my guess is that if we do a simulation where you have a polymer and you pull on one side of the polymer for a certain time scale, you may observe this type of trajectory that you have shown, where you have some part that are described as drift, some part more diffusing, or you can have also a sum of drift plus Brownian motion. That is, you cannot say that this part is Brownian motion, but the two are summed.
Typically, if you do that combination and you get subdiffusion out of it, then you have to have some oscillatory component.
No, not subdiffusion. I mean, okay, what do you mean by subdiffusion?
Alpha was 0.4.
Yeah, I mean, you get alpha, which is something in between a diffusion and drift. So this was our way, actually.
No, this alpha is not between diffusion and drift, is it?
So when you do this, because you have a delta t which is finite, a finite delta t. You don't go delta t goes to zero like in mathematical limits.
No, I understand it. Yes.
And then when you do this, actually the two mix together. And this is how we showed that from this polymer beta model that we proposed, we could only have alpha less than 0.5. That is 0.5, this is Ross. Now how do you describe alpha, which is between 0.5 and 1? We didn't know for a couple of years. And then we said, why don't you impose a force at one part of a monomer, of the polymer, how it affects alpha. And then this was a way to increase alpha because of the finite delta t.
I'm saying it's harder to reduce alpha.
It has to what?
It's harder to reduce alpha. You need to have some kind
No,
of like
to reduce alpha, you need to have all of these forces. To increase alpha, you need, as Pietro said, you need a force. You need a force, something that pull on it with this subpart. So it might be possible to look at your results having a sort of polymer configuration. You pull on it. You look at a certain. You see, if you look, let's do this thinking experiment. If you look at the bead where you push, you just only see deterministic motion. If you look at the bead which is the further away, it should be like more Brownian motion. But in between, you will see the sum, and this maybe tell you where you are in this process of pulling.
So something that we really want to do, but we don't have the strains yet, is experiments where we have 2 or even 3 with different colors positions in the chromosome in the same video, because then we can do cross correlations and we can check the idea that David just said, and we can also do other things. You can also do the cross correlated motion of dots. Anyway, even aside from active events, that would be very informative, depending on the distance along the chromosome, as opposed to the physical distance between them. That could probably tell us a lot about what's giving rise to these motions. I just didn't remark on this one as I went along. So in the same way that we try to disentangle the prefactor as a function of the size of the dot, the alpha that I told you wasn't changing across the chromosome, but it does actually change as a function of the size of the blob. So it goes to 0.4 when the dots are not too big, and then as the dots become bigger, it goes much closer to 0.5, and there's even some evidence about bigger than 0.5. Anyway, so it's a small change, but it seems significant, and these are just all our experiments at different places on the chromosome. So, but again, this may or may not be explained by models of putting a bigger and bigger inclusion inside a polymer.
More questions? Okay, so let's thank Pietro again. Thank you.